Morning, good afternoon, good evening. Part 2 on the overview of the nervous system. Today we'll continue from action potential. Ode Godwin is still my name. So we'll look at action potential. We'll deliberate on action potential. Okay, so we'll start by saying that an action potential is that rapid but temporal change that happen in the electrical membrane of a cell. That rapid but temporal change in the electrical membrane potential of a cell. I want to say particularly the neurons and the muscles. I want to also add by saying that that fundamental process in the functioning of the neurons and the muscle contraction, hmm, action potential is a fundamental process. So without action potential, there will be no impulse transmission and there will be no contraction of the muscles. I want to say there are gates or channels, pumps that are responsible for action potential. One of them is the sodium gated channel or sodium gated pump, and the second one is the potassium channel. Now, these two channels are responsible for action potential. Now, there are some concepts that we need to deliberate for us to understand action potential. One of the concepts is the resting membrane potential. Resting membrane potential. I want to say that before an action potential will occur, neuron must be at its resting stage. Before an action potential will occur, neuron must be at its resting stage. I want to say that the resting stage of neuron is about minus 70 megavolt, and that this minus 70 megavolt is maintained by sodium potassium pump as well as link channel. Now, which one is called the link channel? It is that ion channel that remains open in the membrane of a cell. That ion channel that remains open in the cell membrane is what we call the link potential. I want to also say that there are other concepts that is important even in action potential. One of them is the threshold potential. The threshold potential. I want to say that it is that critical level which the membrane potential must be depolarized. That critical level in which the membrane potential must what? Depolarize in order to initiate an action potential. Or if that is not still okay, we want to say that the threshold potential is typically around minus 55 megavolt, minus 55 megavolt, and it varies depending on the type of cell where the threshold is being generated. We want to also say that it is the membrane voltage. Threshold potential is the membrane voltage at which voltage gated sodium channel open rapidly. So for sodium channel to open, the membrane potential must move from 70 minus 70 to minus 55 before the sodium channel can open. If it is not at that level, it will not open. Okay, so it is for every cell, whether it is the neuron or whether it is the muscle cells, for an action potential to occur, they must move from minus 70 megavolt to minus 55 megavolt. All right, so there is another concept that is important, and that is what we call depolarization. Depolarization. So we are saying that for an action potential to occur, neurons must get to the threshold potential, and that threshold potential is minus 55 megavolt. So at minus 55 megavolt, sodium gated channel will open. And when sodium gated channel open, sodium will flow into the cell. Sodium will flow into the cell. And this negative membrane potential will become positive. And it will continue like that until the membrane potential become about plus 30 megavolt. When they become plus 30 megavolt, what will happen? The sodium gated channel will close and potassium gated channel will open at plus 30 megavolt. Sodium gated channel will close and potassium ion channel will open, and then potassium would flow out of the cell rather. Sorry. Potassium would now flow out of the cell. It will continue to flow out of the cell. And we said at this point, the membrane potential is being restored back to negative. Membrane potential is being restored back to negative. Now, this stage at which membrane potential move from positive back to negative is what we call repolarization, or the process by which potassium gates open and potassium flow out of the cell is called repolarization. So it is restoring the negative membrane potential. And we are saying that sometimes it become necessary for potassium gates to remain open, to open a bit longer than necessary. And when it open a bit longer, it will move beyond the resting potential. So it will move from minus 70 megavolt to minus 80 megavolt. At this stage, we say the membrane potential is called hyperpolarization. Hyperpolarization, where potassium channel remain open a bit longer beyond the resting potential. Beyond the resting potential, we say this is called hyperpolarization. But again, there are other points that we need to look at. That's the refractory period. Now, the refractory period is that period where there is no stimuli. Stimuli cannot occur. So an action potential cannot happen. Or if an action potential or stimuli can be initiated, it must be a stronger than normal stimuli that will occur. So we have two types. We have the absolute refractory period and the relative refractive period. Now, the absolute refractive period is that period where there is no initiation of an impulse and action potential cannot occur. At this point, we say there is inactivation of the sodium gates. There is inactivation of the sodium gates. What of relative refractive period? The relative refractive period is that period where you need greater than necessary or greater than normal stimuli to initiate an impulse, greater than normal stimuli to initiate an impulse for action potential to occur. That is what we call the relative refractive period. Okay, so that takes us to the topic meninges. Let's look at meninges. Very, very important thing to look at in meninges. Number one important thing that we need to look at is that the meninges are membrane, three-layer membrane that are found between the skull and the brain, and between the vertebral foramen and the spinal cord. They are found between where? The skull and the brain, and between the vertebral foramen and the spinal cord. Now, from outside to inside, let's describe it from outside to inside. Once you say that we have the Dura Mater, the Arachnoid Mater, Dura Mater, Arachnoid Mater, and the Pia Mater. From outside to inside, Dura Mater, Arachnoid Mater, and the Pia Mater. Okay, so we want to say that, let's start with the Dura Mater. Want to say that the outermost layer that surround the central nervous system is called the Dura Mater. Now, in the cranium or in the skull, we say that the Dura Mater is double layered, while in the spinal cord it is a single layer. Now, the double layer in the cranium is known as the periosteal and the meningeal layer. The periosteal and the meningeal layer. Now, the periosteal adhere to the skull. It adhere to the skull, while the meningeal layer lie beneath the periosteal and then attaches to the arachnoid mater. Now, this meningeal layer continues up to the spinal cord, known as a single meningeal layer. I want to say that the dura mater is dense, tough, fibrous tissue. Dense, tough, fibrous tissue. That is the dura mater. Then the next important thing we need to look at is the functions, as well as the let's look at the function as well as some features that are very, very important in action potential. One of no in dura mater. In dura mater, what are the functions? Now, one of the function of the dura mater is protection. Want to say that because they lie between the skull and the brain, and between the vertebra foramina and the skull, it acts as protection against mechanical injury. It serves as protection against mechanical injury. What of support? Want to say that there are dura folds that are found on the that are found on the dura mater. One of them is called the falx cerebri. The falx cerebri is that fold that is found between the two cerebral hemisphere. That fold that is found between the two cerebral hemisphere. Then we have the tentorium cerebelli. It is a horizontal fold that separates the cerebrum from the cerebellum. A horizontal fold that separates the cerebrum from the cerebellum. Then we have the falx cerebelli. It is a small vertical fold between the two cerebellar hemisphere. All right, so we have another thing again that is very important, which is the venous drainage. Venous drainage. That's another important point. We want to say there are many venous drainage called sinuses that are found in the dura mater. One of them is the superior sagittal sinus. We also have the inferior sagittal sinus. We have the straight sinus. We have the transverse sinuses. We have the sigmoid sinuses and the cavernous sinuses. All these sinus drain blood from the brain into the internal jugular vein. It drains blood from the brain into the internal jugular vein. Now, I want to say there are other clinical significance in the dura mater. We'll look at them in a short while. These are epidural, subdural hematoma, meningitis, but we will not talk about that in detail for now. Later, as we continue our disorders of the nervous system, we'll look at that point. Okay, so now let's move to talk about the second layer. The second layer is called the arachnoid mater. It's called the arachnoid mater. It is a layer of fibrous tissue. It lies between the dura and the Pia Mater. It lies between the dura and the Pia Mater. It lies between the dura and the Pia Mater. All right. So let's talk about some of the structures that are found in it, in the arachnoid mater. Before then, let's say that the arachnoid mater is a thin, transparent, and web-like membrane, thin, transparent, web-like membrane. Want to also say that there are space between the arachnoid matter. Now, beneath the arachnoid matter lies the subarachnoid space, and subarachnoid space contain the CSF, cerebrospinal fluid. And we'll know the function, but we'll look at it in a short while. But what are the functions of the arachnoid matter? I want to say number one, it acts as protective barrier to the central nervous system. Arachnoid matter act as protective barrier to the central nervous system. Number two, I want to say that it facilitates the circulation of CSF using what the subarachnoid space. The number three, I want to say that it helps absorb shock. Arachnoid matter help absorb shock and maintain a stable environment for the brain and the spinal cord. Okay, so that is for the arachnoid matter. The next important point is the Pia Mater. I want to say the Pia Mater is a delicate layer of connective tissue. It contains many minute blood vessels. It adheres to the brain directly, and then it covers the convocations and deepen into each fissures of the brain, and it continues downward into the spinal cord. That is the Pia Mater. Okay, after the Pia Mater, but before we dive into looking at the next topic we want to deal with, let us look at this diagram. It gives us a clear picture of the meninges, just as we have described now. Want to say here, this place that looks brownish in color is the scarf. That is the scalp. That is the scalp. This place that look brownish here is the scalp. Okay, so when we move from the scalp, we go into the skull. So this is the skull. Here we have the skull. Also, we can call it the cranium. You can see it here, the cranium. Then when we move from this cranium, we'll meet this black line, the black thick line here. The black thick line is what we call the Dura Mater. This is the Dura Mater. Now, between the Dura Mater and the Arachnoid Mater, between the Dura Mater and the Arachnoid Mater is a space. That space is known as the subdural space. Look at it here. It's called the subdural space. After the subdural space, then we'll come to this tiny line here that looks like gray in color. That line is what we call the arachnoid matter. Now, between the arachnoid matter and the Pia mater is a space known as the subarachnoid space. So look at the subarachnoid space. After the subarachnoid space is the Pia mater. You can see it here. This next line that moves into the fissures of the brain, or to the convolutions of the brain. We call it the Pia mater. Okay, so that is the description as we have described. So after that, we move to the ventricles. Very, very important for us to deal with the ventricles. Want to say the ventricles are irregular shapes. Ventricles are irregular shapes that are found in the brain. Irregular shapes that are found in the brain. We call them ventricles. How many ventricles do we have? We have four ventricles. The right lateral ventricles. I repeat, the right lateral ventricles. We have the left lateral ventricle. We have the third ventricle, and we also have the fourth ventricle. So how many ventricles do we have? Right lateral, left lateral, third and fourth ventricle. Okay, so let's take them one after the other. Let's describe this ventricle one after the other. Let's start with the right. Okay, let's just join together the lateral ventricles. Let's just describe them together. Want to say they lie within the cerebral hemisphere. The lateral ventricle lie within the cerebral hemisphere. So the right lateral ventricle is on the right hemisphere, that's the right cerebral hemisphere, while the left lateral ventricle is on the left cerebral hemisphere. Okay, so want to also say that these ventricles, they are separated from each other, that is the right and the left lateral ventricle are separated by from each other by a thin membrane known as the septum lucidum. The septum lucidum separate the right and left lateral ventricle. I want to say that they are lined by ciliated epithelium. The lateral ventricle is lined by ciliated epithelium and they communicate with the third ventricle. The lateral ventricle communicate with the third ventricle through the intraventricular foramina. They communicate. The lateral ventricle communicate with the third ventricle through the intraventricular foramina. Now we move to the third ventricle. I want to say it is a cavity that is situated below the lateral ventricle. The third ventricle is situated below the lateral ventricle. It is found between the two parts of the thalamus. The third ventricle is found between the two parts of the thalamus. I want to say it communicates with the fourth ventricle. The third ventricle communicates with the fourth ventricle through a canal known as the cerebral aqueduct. Through a canal known as the cerebral aqueduct. Then the fourth ventricle. I want to say the fourth ventricle is a diamond shaped structure. It is situated below the cerebellum. The fourth ventricle is situated below the cerebellum and the pons. It is situated below the cerebellum and the pons. It continues below with the central canal of the spinal cord. The fourth ventricle communicates with the spinal cord through the central canal of the, okay, it continues below with the central canal of the spinal cord. It communicates with the subarachnoid space through the foramina in the roof. It communicates with the subarachnoid space through the foramina in the roof. Now let's go back to the first diagram, the last diagram we just deal with, so I can understand the ventricle. Now look at this. This is the lateral ventricle. This is the lateral ventricle. And then lining it is an epithelial cell. Then apart from that, we have the choroid plexus. Look at it here. These are the choroid plexus that has the epididymal cells, right? That has the epididymal cells. Now these are responsible for production of CSF. Okay, so when the CSF are produced, we can see it there. It passed through this opening known as the foramen of Moron. If you want to call it the foramen of Moron, you call it the intraventricular foramen. So either you call it the foramen of Moron or the intraventricular foramen. Now, after that, it moves to the third ventricle. So this is the third ventricle. Remember, I said the third ventricle is found in between the hypothalamus. Then it communicates with the fourth ventricle through the what? Communicate with the fourth ventricle through another opening, and in this time we'll call it the cerebral aqueduct. We'll call it the cerebral aqueduct. So that's where it communicates with cerebral aqueduct. Then from the third ventricle, it moves to the fourth ventricle, which is diamond shape. You can see it here, diamond shape, and this diamond shape communicates with the subarachnoid space. It communicates with the subarachnoid space through the foramen that is found in their roof or in the foot ventricle roof. Okay, so this is our lateral, third, and fourth ventricle as it communicates with each other. So it enables the flow of CSF in from the brain to the spinal cord. Now, from the brain, it flows through the subarachnoid space. You can see it here, and then through here, it can also flow down into our spinal cord. Okay, so that is for the ventricles. Let's move forward to look at the cerebrospinal fluid. Very, very, very important. Cerebrospinal fluid. I want to say this morning that the cerebrospinal fluid is secreted continuously at a range of about 0.5 ml per minute. In a day, about 720 ml of CSF is produced. I will say the volume remained fairly constant at about 150 ml. So it's either it is reducing below that or thereabout. Then it is absorbed. Absorption takes place with secretion. Absorption takes place with secretion. We want to also say that the CSF pressure is measured using a ventricular tube attached to a lumbar puncture needle. We say when you sleep, when you are sleeping or you are lying down, the CSF secretion is about 10 centimeter of water, and when sitting is about 30 centimeter of water. I want to say that the CSF is a clear, colorless, slightly alkaline fluid with a specific gravity of 0.005. Specific gravity of 0.005. Okay. So let us look at the composition of the CSF. Want to say that the CSF is composed of water 99%, electrolytes like the sodium, like potassium, like magnesium is found there. Glucose is also there. Plasma protein, especially albumin and globulin. Few leukocytes are present. Urea is there. Enzymes, and then few of neurotransmitters. Few of neurotransmitters. Now, what is the function of the CSF? And CSF want to say that it support and protect the brain and the spinal cord. It support and protect the brain and the spinal cord by helping them to maintain a uniform pressure. It also act as a cushion or shock absorber between the brain and the spinal cord. It also keeps the brain and the spinal cord moist. It keeps the brain and the spinal cord moist. Then it also aids in exchange of nutrient. It aids in exchange of nutrient and waste product. Finally, it regulates breathing. CSF regulates breathing.